Biochemistry 401 Lecture 25. Today we're going to talk about the pentose phosphate pathway. This is abbreviated PPP. We're going to start with an overview of the pathway, and then we're going to discuss the differences between NADH and NADPH. We're then going to discuss the pentose phosphate pathway in depth, the reactions, and the function of the pathway overall. Now, the pentose phosphate pathway is a very important pathway that is found in the cytosol. It has many purposes. It has two phases, the oxidative phase that is shown in white and the non-oxidative phase that's highlighted in yellow. The oxidative phase is mainly concerned with the production of NADPH, whereas the non-oxidative phase is involved with the interconversion of sugars. The products of the pentose phosphate pathway are NADPH for reductions and ribose for nucleotide synthesis and glycolytic intermediates. These intermediates can go on through the rest of the glycolytic pathway to form pyruvate for ATP production, or these intermediates can be sent back through gluconeogenesis to form glucose again. And so this pathway is very versatile, and we'll see that it serves many functions. In this slide we see NAD+ on the left and NADP+ on the right. And these two are different both structurally and functionally. NAD+ has an OH at the 2' position in the adenine. In NADP+, there is a phosphate group there at the 2' position in adenine. That's the structural difference. Now functionally, they're quite different. The ratios in the cell of NADH and NAD+ favor NAD+. There's more NAD+ than there is NADH in the cell, and so NAD+ is used to oxidize substrates in catabolic pathways. With NADP+, it's very different. The ratios of NADPH and NADP+ favors NADPH, and therefore NADPH is going to reduce substrates, and it does so in reductive synthetic reactions that occur in anabolic pathways. Now, another thing that NADPH does is it reactivates glutathione, and so it's a very important molecule to have in sufficient quantities. And so these two electron carriers serve different purposes. For instance, in the mitochondrial matrix, NAD+ oxidizes substrates to produce reducing equivalents, NADH. This happens in the citric acid cycle, in fatty acid oxidation, and also as we saw in the pyruvate dehydrogenase complex. Then this NADH reduces electron carriers in order to power ATP synthesis. This happens in the electron transport chain. In the cytoplasm, NADPH reduces substrates in reductive synthetic reactions. These include fatty acid synthesis, cholesterol synthesis, nucleotide synthesis, and neurotransmitter synthesis. Now, NADPH is a very important electron carrier, and it serves many purposes. It's used for reducing equivalents in biosynthesis, as we saw just previously. But it also combats infection, and it does so because it's used in neutrophils for the synthesis of reactive oxygen species to make superoxides and peroxides from oxygen in order to kill microbes. Neutrophils are our body's first line of defense in combating infections. You'll often find neutrophils there first. And these white blood cells synthesize superoxides and peroxides from molecular oxygen. NADPH also is important in the synthesis of nitric oxide. Now, nitric oxide is a short-range hormone that also serves many purposes. It can act as a vasodilator by increasing the diameter of blood vessels to allow greater blood flow. It's also used in the synthesis of reactive nitrogen species, and these also will kill bacteria. NADPH is also used to reduce and thereby reactivate glutathione. And this is a very potent antioxidant. Let's take a look at this. Here is glutathione in its reduced state. This is actually a tripeptide that's made up of glycine, cysteine, and glutamate. This tripeptide protects against oxidative stress by reducing oxidized components in the cell. But the thing is, when this happens, glutathione forms disulfide bonds, so you'll have two glutathiones that are bonded to one another through the cysteines. And this is a problem. We have to get rid of that disulfide bond in order to regenerate the two glutathiones that are independent of one another. And so NADPH can be used as a reductant in order to reduce these disulfide bonds and release these two glutathiones from one another. And so it's important to have sufficient levels of NADPH to keep ourselves safe by reactivating glutathione. And this is especially important in red blood cells. Red blood cells do not have mitochondria, and therefore they rely on NADPH because they rely on the pentose phosphate pathway for most of their reducing power. Glutathione is important as a sulfhydryl buffer to keep important cysteine residues in hemoglobin and other molecules in the red blood cell reduced. Glutathione is also important for reducing peroxides and other reactive oxygen species that can damage membranes and other cellular structures within the cell. In this figure to the right, we see Heinz bodies. Now, Heinz bodies are clumps of denatured hemoglobin that adhere to the membrane, and these Heinz bodies are actually a hallmark of oxidative destruction, and so when red blood cells are confronted with oxidative stress, this is what can happen if there isn't sufficient NADPH to reduce glutathione. Red blood cells are especially vulnerable to oxidative stress. And if there isn't sufficient NADPH available to reduce glutathione disulfide to form independent glutathione molecules, then oxidative stress can have a profound effect on red blood cells. And so glutathione is important as a sulfhydryl buffer in order to keep important cysteine residues in hemoglobin and other molecules in the red blood cell reduced. Glutathione is also important for reducing peroxides and other reactive oxygen species within the cell that can damage membranes and other cellular structures. This is a list of tissues that have active pentose phosphate pathways. The pentose phosphate pathway is most active in the adrenal glands, in the liver and testes, in adipose tissues, in the ovary and mammary glands, and in red blood cells. In many of these tissue types, the reason that the activity of the pentose phosphate pathway is so high is because these tissues produce steroid hormones. The liver produces a sterol, cholesterol. In addition, some of these tissues produce fatty acids, and the production of fatty acids requires NADPH. In addition, red blood cells rely on the production of NADPH for the maintenance of reduced glutathione. So now let's look at the oxidative pentose phosphate pathway. The first three steps of the oxidative pentose phosphate pathway are an oxidation, a hydration, and an oxidation. We've seen this before. We've seen it in the last three steps of the TCA cycle, and we're going to see it again when we look at the breakdown of fatty acids. Let's look at this step by step. In step 1, we're going to oxidize glucose 6-phosphate to produce 6-phosphoglucono-delta-lactone. That is shown here. What we've done is we've produced a lactone at carbon number 1. In the process, NADP plus is reduced to form NADPH and a proton. This reaction is catalyzed by glucose 6-phosphate dehydrogenase. This is abbreviated G6PD, and this reaction is the first committed step. And so the enzyme that catalyzes this reaction, glucose 6-phosphate dehydrogenase, is regulated. The second step is a hydration. We're going to hydrate the lactone, and in so doing, we're going to break the chain to form 6-phosphogluconate. We're then going to oxidize this again, but this oxidation is different in that it is an oxidative decarboxylation. The enzyme that catalyzes this reaction is 6-phosphogluconate dehydrogenase. We're going to end up with a 5-carbon sugar and carbon dioxide and NADPH. And so in the oxidative pentose phosphate pathway, we have 3 reactions. Steps number 1 and 3 produce NADPH. And step number 3 produces the 5-carbon sugar, ribulose 5-phosphate, plus carbon dioxide, in addition to NADPH. And so here we see reaction number 1. This is the first committed step in the oxidative pentose phosphate pathway, and it's catalyzed by the enzyme glucose 6-phosphate dehydrogenase. Because this enzyme catalyzes the first committed step, it is a regulated step, and it is regulated by the ratio of NADPH to NADP plus in the cell. A high ratio inhibits glucose 6-phosphate dehydrogenase. This means that there's enough NADPH in the cell relative to NADP plus. And so this pathway is not needed, and it would be better to do other things with glucose 6-phosphate than to use the pentose phosphate pathway. However, when there's low concentrations of NADPH, resulting in a low ratio of NADPH to NADP plus in the cell, this stimulates glucose 6-phosphate dehydrogenase to get the oxidative pentose phosphate pathway in high gear. This means that you're running out of high energy reducing equivalents, and it's a good thing to get this pathway going. In the second reaction, the hydration reaction, we're going to hydrate the unstable ketone to make 6-phosphogluconate. We're going to add water to the ketone at carbon number 1 to form the acid. 6-phosphogluconic acid is rapidly deprotonated to form 6-phosphogluconate. Now we have the second oxidation. This is an oxidative decarboxylation. We're going to take the 6-carbon 6-phosphogluconate and turn it into a 5-carbon sugar, ribulose 5-phosphate. The enzyme that catalyzes this is 6-phosphogluconate dehydrogenase. And the products of this reaction are NADPH, ribulose 5-phosphate, and carbon dioxide. Now if we look at ribulose 5-phosphate, we can see that it is a 5-carbon sugar, and therefore it is a pentose. That's where the name pentose phosphate pathway comes from. This is also a ketose. The first phase of the pentose phosphate pathway is the oxidative phase. We've taken glucose 6-phosphate and we've oxidized it in 2 reactions to form a ketose, ribulose 5-phosphate. Now from ribulose 5-phosphate, we can do 2 things. We can isomerize this sugar to make ribose 5-phosphate, or we can epimerize it to form xylulose 5-phosphate. Now, ribose is an aldose, whereas xylulose is a ketose. And so from ribulose 5-phosphate, shown on the right, which is a ketose, we can use either of 2 enzymes. Phosphopentose isomerase, in which we're going to isomerize ribulose 5-phosphate to form an aldose that is shown on the upper left-hand side of the slide, or we're going to take an epimerase, phosphopentose epimerase, to make another epimer of ribulose 5-phosphate. In this case, we're just making a C3 epimer. Xylulose 5-phosphate is a C3 epimer of ribulose 5-phosphate. Now, ribose 5-phosphate, as you recall, is an important component in nucleic acids, and so we can make a lot of ribose 5-phosphate using this pathway to support nucleic acid synthesis. From this part of the pathway, we can gain fructose 6-phosphate and also glyceraldehyde 3-phosphate to use these in gluconeogenesis or in glycolysis. In the non-oxidative pentose phosphate pathway, we have 3 main reactions that are going to go on to generate glycolytic intermediates from excess ribose 5-phosphate, either from the oxidative reactions of the pentose phosphate pathway or from the diet, from dietary nucleic acid. Now in reaction 1, we're going to use something that's called a transketolase. We're going to take 2 5-carbon sugars and transform those into a 3-carbon and a 7-carbon sugar. In reaction 2, we're going to take a 3-carbon and a 7-carbon sugar and use those to make a 6-carbon and a 4-carbon sugar. Then, in step number 3, we're going to take a 4-carbon sugar from the last reaction and a 5-carbon sugar from another reaction, and we're going to combine those to yield the glycolytic intermediates a C6 and a C3 carbon sugar. The C6 and the C3 carbon sugars that we generate in the end are fructose 6-phosphate and glyceraldehyde 3-phosphate, respectively. And so the overall scheme of this portion of the pentose phosphate pathway is we're going to use 3 ribose 5-phosphate molecules to yield 2 fructose 6-phosphates and 1 glyceraldehyde 3-phosphate. This is reaction number 1. We're going to take a transketolase and we're going to transfer 2 carbons from xylulose 5-phosphate to ribose 5-phosphate. And we're going to be left with an aldehyde, glyceraldehyde 3-phosphate, and a ketose, sedgeheptalose 7-phosphate. So what we've done is we've changed xylulose, which is a ketose, to an aldose, glyceraldehyde 3-phosphate. And we've changed an aldehyde, ribose 5-phosphate, to a ketose, sedgeheptalose 7-phosphate. Now the transketolase enzyme has a problem, and that it can only use substrates if the C3 carbon has a configuration of xylulose, and that's why we had to epimerize ribulose 5-phosphate to make xylulose. And so at the end of this reaction, we have a 3-carbon sugar, glyceraldehyde 3-phosphate, that is an aldehyde, and a 7-carbon sugar that is a ketose. Now in this reaction, we're going to take the products from the previous one, glyceraldehyde 3-phosphate and sedgeheptalose 7-phosphate, and this time we're going to transfer 3 carbons. So transaldolase transfers 3 carbons. We're going to take 3 carbons from sedgeheptalose 7-phosphate, and we're going to add those to glyceraldehyde 3-phosphate. In the process, we'll be left with fructose 6-phosphate, which is one of the products that we want, and erythrose 4-phosphate. And the enzyme that catalyzes this reaction is called a transaldolase. We're going from an aldose to a ketose, glyceraldehyde 3-phosphate to fructose 6-phosphate. And we're going from a ketose, sedgeheptalose 7-phosphate, to an aldehyde, erythrose 4-phosphate. Now reaction number 3 is catalyzed by a transketolase again. Transketolases transfer 2 carbons. So we're going to go from an aldose and a ketose to a ketose and an aldose. We're going to take the 4-carbon erythrose 4-phosphate and the 5-carbon xylulose 5-phosphate, and we're going to transfer 2 carbons from xylulose to erythrose. We're going to be left with fructose 6-phosphate and glyceraldehyde 3-phosphate. And so with this reaction, we have the 2 other glycolytic intermediates. So from 3 ribose 5-phosphates, we can gain 2 fructose 6-phosphates and 1 glyceraldehyde 3-phosphate. And this is shown in the diagram here. And the arrows indicate where these products are made in respect to the rest of the non-oxidative pentose phosphate pathway. Please recall that glyceraldehyde 3-phosphate enters glycolysis in this portion in stage number 2, whereas fructose 6-phosphate enters glycolysis in stage number 1 at this point. Now the thing is that there are different ways to run these pathways depending on the needs of the individual. There are some times when you're going to need a lot of ribose 5-phosphate to make nucleotides. There are other times when you're going to need a lot of NADPH because you're doing synthetic reactions like steroid synthesis. There are some times when you're going to need both NADPH and ribose 5-phosphate. And there are some times when you're just going to need a lot of ATP. And so the different ways to run these reactions are classified into 4 different modes. In mode number 1, we have a cell that needs much more ribose 5-phosphate and not so much NADPH, Maybe these are rapidly dividing cells that need a lot of ribose 5-phosphate to support nucleotide synthesis for DNA synthesis in replication. In this case, we're going to take products from glycolysis to make ribose 5-phosphate. We're not even going to worry about the oxidative pentose phosphate pathway. We're just going to go from fructose 6-phosphate and glyceraldehyde phosphate and run the reactions of the non-oxidative pentose phosphate pathway backwards to make ribose 5-phosphate. And that's what we see here. We're going to take glyceraldehyde 3-phosphate, indicated at the bottom right-hand corner of the slide, and 2 molecules of fructose 6-phosphate. And we're going to run the reactions of the non-oxidative pentose phosphate pathway backwards to yield ribose 5-phosphate and xylulose. The ribose 5-phosphate we're going to use to make nucleic acids. And the xylulose we can use to yield ribose 5-phosphate. Now there are some cases where you'll need a lot of NADPH and a lot of ribose 5-phosphate, both at the same time. And so to do this, we're going to use the oxidative pentose phosphate pathway. And once we get finished with the oxidative phase, we're just going to siphon off the ribose 5-phosphate and not even going to worry about the non-oxidative pentose phosphate pathway at all. Now, mode number 3 is where you need a lot of NADPH and not so much ribose 5-phosphate. And this happens when you're going to do a lot of steroid synthesis or a lot of fatty acid synthesis. In this case, you're going to use both the oxidative and the non-oxidative pentose phosphate pathway. You're going to send the products through gluconeogenesis to make more glucose 6-phosphate. So, what we're going to do is we're going to run both parts of the pathway. Then once we get glyceraldehyde 3-phosphate and fructose 6-phosphate, we're going to send those back up gluconeogenesis to make more glucose 6-phosphate. So we're going to take glucose 6-phosphate and we're going to send that through the oxidative pentose phosphate pathway to make ribulose 5-phosphate. We're then going to take ribulose 5-phosphate and use that to make ribose 5-phosphate. We're going to use that in the non-oxidative pentose phosphate pathway to make fructose 6-phosphate and glyceraldehyde 3-phosphate. And we're going to send those up through gluconeogenesis to make more glucose 6-phosphate so that we can generate NADPH and ribose 5-phosphate. So what happens when you need NADPH and energy? So you're not going to be able to take glyceraldehyde 3-phosphate and fructose 6-phosphate and go through gluconeogenesis because you need ATP, and gluconeogenesis takes a lot of ATP. It is an energy-intensive process. So what you're going to do instead is you're going to take glyceraldehyde 3-phosphate and fructose 6-phosphate, and you're going to send those down through glycolysis to generate ATP and the production of pyruvate and more ATP in the TCA cycle and in oxidative phosphorylation. Now, the regulation of the pentose phosphate pathway occurs through monitoring of the NADPH to NADP plus ratio. The reaction that's regulated again is the one that's catalyzed by glucose 6-phosphate dehydrogenase. This is an essentially irreversible step, and it is also rate limiting. It is also the first committed step of the pentose phosphate pathway. And so this is the point of regulation in the pentose phosphate pathway. This enzyme is regulated by levels of NADPH to NADP plus in the cell. Now the reactions of the non-oxidative phase are also regulated, but they are regulated at the level of the substrate availability. If there's a lot of substrate around for the reaction to occur, then the reaction will proceed. A deficiency of glucose 6-phosphate dehydrogenase is the most common enzyme deficiency that's seen. This is caused by X-linked genetic mutation, and so it is most commonly seen in males, but can also be seen in females. The thing is, usually people don't even know that they have this deficiency. because usually persons are asymptomatic. This disease is usually subclinical, having no outward symptoms usually. However, symptoms can arise when a person is subjected to oxidative injury. When this happens, the person will get what's called hemolytic anemia. And this is an anemia that's caused by the premature rupturing of red blood cells. When red blood cells cannot maintain membrane integrity, they will be more prone to rupture. And so hemolytic anemia is an anemia that results from the lysis of red blood cells. This happens especially in response to an oxidative challenge, and we see these symptoms because in red blood cells, the pentose phosphate pathway is the only source of NADPH, and NADPH is very important in order to keep glutathione in a reduced state, and therefore keep other molecules in a reduced state. Generally speaking, the environment of our cells is a reducing environment, and the way that we keep that, a reducing environment, is through the action of NADPH and glutathione. This makes sure that disulfide bonds do not form where they're not supposed to. When there isn't enough NADPH in the cell, we can have disulfide bonds forming in really bad places. This causes a misfolding of proteins such that you can have hydrophobic spots being exposed to the aqueous environment. This can cause protein clumping, and it can also cause proteins to adhere to membranes when they're not supposed to. Other things that can happen is that you can also have free radicals being formed that cannot be reduced by NADPH because there isn't enough available. And oxygen radicals are highly destructive, especially to membranes. And so if red blood cells cannot maintain the integrity of their membranes, they're far more apt to rupture. And this is where you see the symptoms in hemolytic anemia. Now, these symptoms can arise when a person has what's called an oxidative challenge. This means that they're exposed to those things in the environment that have an oxidative outcome. This can be oxidative drugs such as some antibiotics, some antimalarials, and some fever reducers. Other oxidative challenges include the ingestion of fava beans and infection. Now, a hemolytic anemia that's precipitated by fava bean ingestion is called favism, and we'll talk about this in just a minute. Infections are also a source of oxidative challenge because, as you'll recall, neutrophils produce and release peroxides. These peroxides are highly reactive oxygen species, and if there's not enough NADPH to combat this, the cells that are exposed to these peroxides, the healthy cells, can also be damaged. Now, another clue that there is a deficiency in glucose 6-phosphate dehydrogenase is a prolonged neonatal jaundice. This is usually seen in the first 4 days after birth, and again, it's caused by an increased susceptibility to rupture of red blood cells. Favism is a disease that is usually seen first when a patient eats a lot of fava beans. Now, fava beans are also called broad beans, and they differ from green beans. They're a completely different species. These are a main staple in many diets. They're seen in Africa and along the Mediterranean. And they contain active oxidants, which include visine and convisine. Now, the thing is, not every person who has a deficiency in glucose 6-phosphate dehydrogenase has favism. So that there are some persons who can ingest fava beans with no problem because they do not have a specific mutation in the dehydrogenase that will respond to this. Now, one of the reasons that one sees a lot of deficiencies in glucose 6-phosphate dehydrogenase is the fact that it is well tolerated. The symptoms are usually not present, so there usually isn't a problem, unless, of course, a person is exposed to an oxidative insult. But under normal conditions, a person doesn't even know that they have a deficiency. However, there are some benefits of having this deficiency. And you'll see a high prevalence of this deficiency in areas where malaria is endemic. And it is thought that this is because this particular deficiency affords some protection against death from malarial infection. And so persons who have this deficiency are able to live to reproductive age. And this marks the end of Lecture 25. Have a great day.